Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing in continuation with ICH S9 question and answer guide question and answer guideline. ICH S9 guideline question and answer document part 4 will discuss. So we'll discuss the questions with respect to other considerations. So let's see question number 27. Question number 27 is under section 4.1 of the guidance states, means ICH-9 guidelines states that safety of the conjugated material is the primary concern. So if you look into this anti-drug conjugate, is there no ADC, it consists of three parts. One is antibody, then you have payload as linker. Okay, this conjugated material is, its safety is a primary concern, it is saying, and the safety of the unconjugated material. What is unconjugated material? Here it is linker or payload. Okay, can have more limited evaluation for an ADC. Means together all these three A plus B plus C. What does a more limited evaluation mean? So in the guideline it says that limited evaluation is enough in the case of unconjugated material. So the requester is asking to explain in detail. So what is the regulatory agency ICH is stating here? We'll see. Here they are clearly defining what is this unconjugated material. The unconjugated material in section 4.1 of ICH S9 guidelines refers to the payload. So you have to understand what is unconjugated material. It is here payload. Okay, drug which is used to fight the cancerous cells. Okay, cytotoxic payload here. Okay, the whole antibody drug conjugator should be tested in at least one species, whether it is dog or monkey, relevant species. See question number 29 for the discussion of the payload. We'll see now we are discussing question number 27. So before going that, try to understand, as I told you, this is the complete structure of antibody drug conjugate. These are different components, antibody, linker as well as payload. Here you can see how at various stages this mechanism, how this ADC mechanism is there, how this vesicle-like formation you can see with respect to payload. Okay, before going into question number 28, let's very quickly go to what is ADC, because not all of us will be working on biopharmaceuticals, so these terminologies might be little bit new to the some of the toxicologists. And also we have discussed about what is anti drug conjugate in the part 3. Also we have discussed about immunoagonist also. Please check section part 3. But quickly we'll revise what is ADC here. Antibody drug conjugates are a class of targeted cancer treatments. Okay, these are used to for the cancer treatment that combine the homing ability of this monoclonal antibody. If you see, this is a typical structure of antibody where you have, this is the heavy chain and these are the light chains, and here is the antigen binding sites. Okay, this is your variable region. Okay, so this monoclonal antibody, no, it is having a tendency called homing ability. Combined with homing ability of monoclonal antibodies with the cell killing power of this chemotherapy. Okay, basically in simple language means the targeted therapy will be the this is the cancer cell. Your ADC will go enter into this cancer cell tumor cells, and it will cause the lysis. It causes the death of this tumor cells without affecting the adjacent or healthy cells. Okay, by delivering potent toxins directly to the tumor cells while sparing the healthy tissue. ADC improve treatment efficacy and minimize the side effects compared to the traditional systemic chemotherapy like that of chlorambucil or taxol derivatives. No, these are more potent and more they are sited delivery are there. So thereby they will not affect the healthy cells. Example we have trastuzumab, which is used in the case of breast cancer. So it is not affecting other cells, only it is affecting breast tissue. Targeted drug delivery can be done. Next, we'll see question number 28. If the antibody of an ADC has not been separately characterized, if this in the case of ADC, one of the component will be your antibody. If it is not separately characterized, should an arm of antibody only be included in the toxicology study? Generally, for monoclonal antibody, separate toxicology study is not recommended by the agency. Let's see what the answer given by the agency. In general, studies of the monoclonal antibody alone are not warranted. This is what the agency says, ICH agency. Now let's see question number 29. Question number 29. Are studies with the payload and linker only recommended? Previous question was only about antibody. Now they are asking, is separately a payload study or a separate linker study is really needed? That is the question. What is the agency answering? We'll see. Before going the actual study, we do some pilot study, no? So the pilot studies and the nature of the payload will determine what additional toxicology studies or pharmacology study or drug delivery studies, if any, are appropriate with the payload or payload linker. Okay, so from the pilot study, initially you will be doing some pilot studies. So from that only you will be needing, but you will be getting an information whether additional studies need to be done or not. Evaluation of the linker alone is not usually warranted. So in the previous question, you see, evaluation of antibody itself, monoclonal antibody itself is not required. So similarly here, linker is not separately doing a tox study on linker is not recommended by the ICH agency. If the toxicity of the payload or payload with linker, whether just payload or payload plus linker, um, has been already characterized in the case of pilot study. So in the case of actual studies, a GLP study of the payload, okay, or payload with linker has not been, may not be warranted or could further be abbreviated or can be waived off. Okay, so if you have a pilot study where it has been already been, we are able to get an information from that pilot study. So further separately studies are not needed. If GLP studies are not needed. If the toxicity of the payload or payload plus linker has not been characterized in previous studies, the payload or payload linker could be evaluated in one species, whether it is monkey or dog, as a standalone study, or could be added an arm into the toxicology study of this. So completely ADC, like antibody linker and payload together, no, you will be doing some one toxicology study, whether 28-day, 3-month or 6-month study you will be doing, no, in relevant species. In that only you can add an additional arm, and that additional arm should contain this payload or payload plus linker, where you can characterize the toxicology of this, particularly this payload or payload plus linker, if there is any toxicology associated with this payload or payload plus linker group. Okay, so what is agency is suggesting under question 29 only? See also note 2 of ICH S6 R1 guideline. So whenever you are reading such guidelines, no, any ICH series or GMP guidelines will be there, no, regulatory guidelines in, they always cross refer you. Please refer or see, they will give you in the form of notes or end notes or the, so please you have to read. So here in this they have suggested us to go back and refer to ICH S6 guideline, which is a biotechnology related guideline, and they're asking us to read note 2. So let's quickly go through what note 2 is stating and how it is related to ICH S9 and this particular question. If two species have been used to assess the safety of this ADC means all these three together, an additional short-term study or an arm in a short-term study should be conducted in at least one species, whether rat or whether a monkey or rabbit or rat, or with unconjugated toxin. Unconjugated toxin means previous slide we are seeing payload. Okay, in these cases, a rodent is preferred unless the toxin is not active in the rodent. Generally, rodent study is enough, but in case rat is not the relevant species, rat or mice, then if only one pharmacological relevant species is available, for example monkey or a dog, then ADC should be tested in that particular species. A novel toxicant calls for an approach to species selection similar to that of ADC. If there is a new novel toxicant, how you are going to decide in the case of ADC what is the relevant species? By doing certain pharmacological studies whether receptor binding is there or not. Similarly, for this novel toxicant also you have to do it the same approach. For example, in the case of novel excipients, how we will do for the conventional drug molecules, if it is a new excipient, you have to consider as a new chemical and you have to do all battery of toxicology tests. So similarly, it is saying the same thing. A novel toxicant calls for an approach to species selection similar to that used for new chemical entity on a case-by-case approach. Example for anti-cancer products in accordance with ICH S9 guideline. For toxins or toxicants which are not novel, already well known in some other ADC drug it was used, then in such cases no need of doing again toxicology studies. Not novel and for which there is a sufficient body of scientific information available, separate evaluation of this unconjugated toxin means payload is not warranted. Data should be provided to compare the metabolic stability of the ADC in the case of in animals, uh, with humans. Okay, so metabolic stability additionally that is needed. I think it is a bit confusing as of now, right now, because these are not, as I told earlier, not all of us will be working on antibodies or this biopharmaceuticals. So, though, but still, we'll be getting questions on ICH S6 guideline, you'll be getting questions on ICH S9 guideline. I'm telling you, you'll get questions on even on S12 or S11, S8, these type of questions, all ICHS series, either from S1 to S13, some or other questions they will be asking you in the exam. Okay, now we'll go to question number 30. Okay, what toxicokinetic analysis should be performed? Now the question is respect to TK. Should the free antibody and free payload as well as free antibody be distinguished from that of the ADC? So agency answers is current best TK practices for ADC are to measure the level of this ADC completely and the payload and an estimate of amount of the free antibody. Once this is released from this conjugate antibody, this free antibody should you have to measure. Okay, amount of free antibody should be provided. So in the case of TK, you have to measure level of ADC, level of payload as well as level of this free antibody in the case of plasma. Okay, next question. So one question with respect to TK. So now next question is, should plasma stability be included as a part of FIH study plan? So first in human dose, whatever the phase 1 study plan will be there, should we include plasma stability data? If not, at what stages of development is it needed? Whether it is later stages of phase 1 or later or in the phase 2 or phase 3, when this data is needed. So what is the agency is answering is, ICE states that in vitro data about plasma stability of ADC in human and toxicology should be species should be available to support the first in human trial. So this TK data, plasma stability data, the concentration of the free antibody, all this data need to be available at the time of first in human uh, uh, dose. Okay, before starting initiating first in human, you need to have this data. Next question is with respect to FIH only. Is there a recommended approach to to setting an FIH starting dose for ADC? So this is this kind of question you can get in your exam. So for anti-drug, so they will ask you question like this. You're a toxicologist in certain organization, so you are developing one new molecule, which is the nature of that molecule is ADC. So what type of study ICH guideline you need to do, and what kind of toxicology studies that is needed, and they will ask you how you are going to select the first in human dose for this ADC. So let's see what is the answer given by the ICH agency. A starting dose for use in cancer patient should be consistent with the ICH S9 guideline. Since we are discussing ICH S9 here, and those anti-cancer drugs which are used for late stage persons who are severely affected and are late stages of their survival are there, no, for them ICH S9 guideline you have to consistently you have to follow. For example, for the cytotoxic payloads, this one, okay, payload itself is causing cell toxicity. The starting clinical dose can be determined using either 1 by 10th of the severely toxic dose. So in the case of conventional molecules, generally what you will do, you will take a NOEL, you go for fractionation of that one, converting it to HED, then you will be converting it to MRSD, maximum recommended safe dose. But in the case of anti-cancer drugs, the objective is not to derive NOEL, but the objective is to identify STD, that is severely toxic dose in the case of rats. Okay, that is 10%. The definition will be it is a severely toxic dose in the 10% of the experimental population. Death is not there. Okay, and in the case of non-rodents, like in the case of dogs, either in the case of monkeys, so what we are going to identify is, instead of NOAEL, we are going to identify highest non-severe toxic dose, HNSTD. And here the fractionation is 1 by 6 of HNSTD will be considered as the starting dose for this ADC type of drugs. And you can see why there is a difference. Why in the case of STD, rodents, it is 1 by 10th, as well as in the case of mammals, that is non-rodents, monkeys and dogs, it is 1 by 6th, because it is assumed that biologically these dogs as well as monkeys, they are closer to humans, like mammal. For that reason, they are using the uncertainty factor. They are decreasing it to 1 by 6, assuming that there is not much biological variation within the mammals. So they are going for lower factor, that is 1 by 6, and we are considering HNSTD. Whereas in the case of rats, because of there is a large variation between interspecies variation between the rat as well as human. So we are going for higher factor, that is 1/10th. Okay, and it is severely toxic dose. So for to address that correction, you are going for higher factor 10. So one question you have to, as a risk assessment toxicologist, you have to remember why 1/10th and why 1/6, why STD for rodents and why HNSTD for non-rodents, why NOEL for the conventional molecules, as well as why HNSTD for in the case of anti-cancer drugs. Okay, for the so coming back to the question, for the ADC based on body surface area, BSA is very important, because in the case of cancer treatment drugs, the base for considering or deciding the dose is not body weight, because in the case of cancer patients, because of the highly severity of the treatment as well as the nature of the cancer cells, no, the body weight is not constant. The cancer patient body weight will be fluctuating. It will become severely, the patient will be severely become decrease in body weight. So body weight won't be considered as a constant to decide the doses whenever the physician is making the treatment options. Then only they will go for BSA, body surface area. And also there are certain cases, if in case there is an increase in tumor cells, there are certain types of tumors where increase in the tissue and the localized tumor will be there. So in that cases also increase in body weight will be there. So in either cases body weight is not constant. That's why BSA is considered as a baseline to derive the doses for the treatment doses for these cancer patients, depending on which is the most appropriate or the sensitive species. So we need to identify. So whenever these anti-cancer drugs we are working, we have to identify the most relevant species. The receptor these anti-cancer drugs are going and binding in the tested species, they should also have those receptors. Okay, other approaches can be considered for new classes of anti-drug conjugator. So hope from this little bit we have discussed about FIH, first in human dose, how it is important, how TK is important, that we have discussed in this slide. Okay, now we'll go to question number 33. So 33 is with respect to cytotoxicity. We'll see, given the extended half-life of an anti-drug conjugate as compared to a cytotoxic small molecule, is a single dose toxicity study used an ADC sufficient to support a clinical dosing schedule of once every 3 weeks? So how to understand this particular thing? If the treatment schedule is only once in every 3 weeks, means on day one you are doing, after day 21 or 22 only second dose if it is there. Second means in the 3 weeks only one time dosing is taking place. So in such a cases, single dose toxicity study in the case of ADC, whether it is sufficient or not, ICH agency, my answer will be yes only. Let's see, at least 2 doses of ADC. Okay, so more than one dose, at least 2 doses of ADC should be administered to support initial clinical trials of once every 3 to 4 weeks. So in this case, 2 doses are recommended for this in that table. So go back and check the part 3 video. There is a table very clearly given by the ICH S6 guideline, what kind of treatment schedule and what kind of non-clinical studies are needed, they have given. So please go and read. Next, if the ADC does not bind the target in the non-clinical species, you have an anti-drug conjugate, new anti-cancer drug, and whatever the species, tested species, in that receptor is not only there, it is not going and binding. What repeat dose in vivo toxicity should be needed? So we don't have a relevant species, does not bind the target in the non-clinical species. So let's see what is the agency, ICH agency is answering. If the epitope is not present in the non-clinical tested species, for example, it is not there in rat or rabbit or monkey or dog, a toxicology study in one species for the ADC should be sufficient. If out of all this, mice, rat, rabbit, dog, monkey, at least it is there in the dog, in one species it is enough. That is what the agency, ICH agency is saying. Alternative models such as transgenic animals or use of homologous molecules is usually not warranted. Out of all these species, in at least one relevant species is there, that is enough. Please do not go for transgenic animal models or tissue models or homologous models. No need of conducting. That's what the agency recommends, ICH agency. Next question is, what is the utility of this tissue distribution studies? So with respect to anti-drug conjugate, so some questions with respect to biotechnology you will be getting. whenever you're developing biopharmaceuticals, no, one section you have to concentrate on tissue cross reactivity. So here they're discussing about tissue distribution studies. So what is the use of this tissue distribution studies? So we have seen in the previous slide about TK, plasma stability. Now we are discussing about distribution. So let's see. And previous question we discussed about half-life also and receptor binding also. How you can see how it is connecting all PK sections, absorption, distribution, receptor binding, okay, elimination, half-life. So agency, research agency states that in general tissue distribution studies of ADC are not recommended or not warranted. So it is not, for ADC type of drugs, tissue distribution studies are not needed. But I will pause the video and go back and read about tissue cross reactivity. There are certain studies called tissue cross reactivity with respect to biopharmaceuticals, so please go back and read. Now come back to question number 36. Generally two species are used for toxicity testings. Okay, generally one you will go for rodent and one you will go for non-rodent, that is rat and your dog or monkey, like that. So generally two species are used for toxicology testing. For an ADC, anti-drug conjugate, are there situations where one species may be acceptable? So similarly, it is, the question is similar to that of 34. Yes, as per my understanding, it is acceptable if there is only one relevant species. Agency will accept, ICH agency, or you are submitting the data to FDA also, they will also accept. So let's see, when the antibody portion of the ADC binds only to human and non-human primate antigens, either in the case of monkey or different species of monkeys will be there, conducting a toxicology evaluation with ADC in only NHP, that is non-human primate, the only relevant species would be appropriate as discussed in ICH S6R1. So you go back and see this section, what kind of tested species will be given in the ICH-S6, that is biotechnology derived guideline revision 1. So there is a statement where they clearly say one species, one relevant species is enough. Okay, there's no point in conducting the study in the case of non-rodent. For example, sorry, in the case of rodent, you are giving the drug, but in the rat that receptor is only not there. There's no point of conducting those studies. For the payload, see response in question number 29. So what is payload? What is unconjugated protein? Separate payload studies, toxicology studies are not needed. You can add an arm to the ADC, typical ADC toxicology study. You can keep a separate group for payload or payload plus linker. Okay. Next, we'll see question number 37. Question number 37 is for metabolites that are human specific. So you're working on an ADC or new anti-cancer drug, and that is you given to a human being, and a metabolite is formed. And metabolite is if it is active, and for example, that metabolite can only seen in the case of human. Relevant species is monkey or dog. In that metabolite is not at all formed. So how to handle that we'll see. So for metabolites that are human specific or present at disproportionately higher levels in the case of humans. So even though if our relevant species is dog or monkey, in that some fraction of metabolite might formed, but that is not having any higher significance when compared to toxicology species, what toxicology evaluation should be done. So for this metabolite, whatever this active metabolite, whichever it is formed specifically in the human, and that too at higher concentrations. So because it was not found in animals, so we didn't do any additional studies to support that metabolite, the importance of, or if that metabolite is causing any toxicity. So it is only forming in the human, that too at higher concentrations. So in such cases, how to qualify that metabolite? What the ICH agency states will see, in general, additional studies with disproportional antibodies are not needed. As per ICH S9 guideline, it is not needed. But what additional statements it states will see, in cases where the metabolite is not produced in the toxicology species. For example, our model was dog, and a relatively high amount of human exposure due to the metabolite. Metabolite is also active, your API is also active, so overall concentration of the drug in the plasma or in the blood or serum will be increased. So unnecessarily the person will be exposed to higher concentration of the drug, right? So and not the API. Additional toxicology evaluation of human metabolites may be considered. So you have to do additional active studies for this active metabolite, which is only seen in the case of human, to qualify that human. Why means I told you, because unnecessary additional exposure will be, the overexposure will be there. So overexposure means over risk will be there. So to avoid that risk, we need to submit, that is will be a agency requirement, either it is from the FDA or EMA or ICH requirement it will be. So one more thing before going to the next question number 38. this is a table given in the ICH S9 guideline only. So what now next two slides we will be discussing about the impurities, how to handle impurities in the case of ICH S9 guideline, and we'll be correlating whenever we are reading ICH S9, ICH M7 as well as ICH Q3A as well as Q3B guideline. So here they have given one table how to qualify the genotoxic impurities. Very quickly I'll run through. So first row it says you are working on certain API, anti-cancer drug. If that API is genotoxic, okay, so and in that API some impurity is coming. If the API is genotoxic, yes, and the impurity, whatever the impurity which you found in this API is impurity exceeds the ICH Q3A or Q3B guideline. There are certain in the Q3A, Q3B you will be having identification threshold, qualification threshold. Okay, so it is exceeding that one qualification threshold. It is not exceeding, no proposed action. Then there is no action need to be taken by that particular scientist or toxicologist. If the API is genotoxic, yes, whatever the experimental drug I am doing, that is genotoxic, and the impurity will exceed the ICH Q3A, Q3B limit, then also there is no need to do any action, because API itself is genotoxic. If the impurity is genotoxic or not, if it is a part of this one only, there is no need to take any action. Third scenario is if the API is genotoxic. If, no, in third case is that API is not genotoxic, it is non-genotoxic, it is not causing any DNA damage, and your impurity which is found in this API, that is also not causing any DNA damage. Both API are not causing DNA damage, your impurity is also not causing any DNA damage. Then in that case, you need not take any additional action, and it is not crossing that concentrations more than a 5HQ3 or Q3B limit. Next one is if the API, whatever the experimental drug, anti-cancer drug is there, it is not genotoxic. It is API is non-genotoxic. Experimental drug is not causing any DNA damage, but your impurity is causing DNA damage, and it is above the ICH Q3A, Q3B threshold limits. Then in such cases, that impurity need to be additionally assessed. You have to risk assess. You have to establish the safety of that impurity. Genotoxicity assessment of impurity should be considered. Either you will go for AIMS essay. In that also again you will be proving. Then you will be going for other two battery of toxicology genotoxic test like micronucleus assay and chromosome aberration. And if it is severely exceeding the limit, then you have to go for a 14-day tox day or like that. You have to establish an OL and you have to derive permitted daily exposure. So that is the background behind this particular table. Pause and go through. Those who are working on drug discovery, those who are working on impurity qualification, they will be well familiar with this particular table. Those who are new to this also, you whenever you're working on a certain drug, compare this table where my particular case will fit into, whether the first one or second or third or fourth. Based on that proposed action you can take. Okay, now we'll go to question number 39. So what is this question number 39? As I told, next two slides we'll be discussing with respect to impurities. So what is this question? Is ICH M7 giving guidance for the management of mutagenic impurities? So ICH M7 guideline is specifically for mutagenic impurities, applicable to the patient population covered under ICH S9. See, ICH S9 is specific. That's why all these guidelines, no, you should be well aware of what is the scope, what is it, to which type of case this my guideline is applicable, and which type of case this guideline is not applicable. If you know these things, then it will be very much easy. Okay, so the scope of ICH M7 specifically states that the guidance does not apply to drug substances as well as drug products, API as well as finished products intended for advanced cancer indications as defined in the scope of ICH S9 guidelines. So M7 is not applicable, or its impurity qualifications, it is not applicable for those drugs which are used for the advanced cancer treatment. Okay, ICH M7 is out of scope. Okay, therefore mutagenic impurities in products used for treatment of indications under the scope of ICH S9 should be considered for management consistent with the concepts outlined in ICH Q3A and Q3B. So whatever the threshold, qualification threshold, identification threshold, whatever they are given in Q3A, Q3B guideline, that you have to follow. Mutagenic impurities in the product used for treatment of indication under the scope of ICH should be considered for management consistent with the concepts outlined in ICH Q3A, Q3B. So next question we'll see question number 40. given the compressed development timelines, because in the case of ICH-S9, the patient is almost about survival time is less, so there is a compressed developmental timelines for these oncology products. Drug substance manufacturing processes may not fully mature. Okay, so complete life cycle is still not completely developed. Batch scale is not completely developed at the time of making the marketing application. Okay, so if new impurities are observed and they are above ISH Q3A, Q3B, for the initial stages, no, they didn't find these impurities. But once they went for scale up, then they identified these impurities. Okay, and they are above ISH Q3B, Q3A, Q3B, the qualification threshold. Now, qualification thresholds after the completion of registration toxicology studies, how should such circumstances be handled? So, situation you understood. There is a case, there is a drug which we are developing during initial stages, small scale manufacturing when you are doing, you didn't identify the product. Once you go for scale up, then you found that there are certain impurities, new impurities have been identified, and those new impurities are also above the qualification threshold as per ICH Q3A, Q3B. Now, how to handle this type of impurities? We cannot just discard the batch. How to handle? What is the answer given by the agency? Will see ICH Q3A, Q3B gives some flexibility to qualification thresholds for impurities under such circumstances. If such a case, so there is some answer given within the scope of this guideline. Let's see what it is. A risk assessment should be considered. So we need to, before immediately jumping into a new type of study, it is the responsibility of the toxicologist as well as the scientist, chemistry person also, to do a risk assessment of what, of already available information. Okay, considering factors like structural similarity to the parent drug. Now you have identified certain impurity, try to compare it with the parent API. Is there any similarity between this API? Then you can go back to the previous table we have seen. No, based on that you can do. Next, toxicology alerts in the case of structure. So whatever the impurity which you have identified, are there any special groups, special motifs like CO group or CHO group or ketone group or something like that, which is responsible for causing toxicity? If we are able to identify that toxicology motif, that functional group, then next step we can take. Then presence of the impurity of lower levels in toxicology or clinical lots. So if we are able to able to justify stating that whatever the phase 1 or phase 2 trials, whatever we are doing, the batch which was used for these trials, no, it contains lesser quantity, lesser percentage of impurity when compared to the current commercial batch. Then metabolite status, is it forming any, is the impurity similar to that of any metabolite which is formed, or what is the percentage that you have to see? Patient group and dosing regimen. So what kind of, whether you're, is there a change in the regimen, like from the advanced cancer to the long term cancer survival patient, or it is for the adult patient or female patient or pediatric patient or geriatric patient or kidney failure patient or liver failure patient? Like that you have to consider these things also, to consider whether in vivo qualification studies should be considered. Such studies may not be necessary in cases just because an impurity is found above a specified ICH Q3A, Q3B. So ICH guideline, ICH agency states that generally new studies are not needed to for this identified impurity. You can go for risk assessment. There are 6 to 7 methods are there where you can, by justifying, by comparing these things, you can justify the agency. When the product is being developed under ICH S9, identifying a novel in the qualifying study is usually not warranted. So additional study, toxicology study, systemic toxicology study, and identifying novel in such cases is not needed. So this particular question is 40 is in more detail, but you will come across this is very frequently in your whenever you are going for scale up from the smaller pilot batch to commercial batch, whenever you, so this type of scenario will definitely come across in the drug discovery. Then next we'll see question number 41. So in continuation of impurity only, one more question is there. If a drug with an impurity is first developed in patient with late stages disease, so I am developing an anti-cancer drug and I first develop it for a cancer treatment for which the survival is only 2 months or 6 months, right? And later moves to a different population. Now I am using it for some colon cancer or pancreatic cancer where the person or liver cancer, the person can survive up to 1 year or 2 years, okay, with long expected survival. Example, those administered pharmaceuticals on a chronic basis to reduce the risk of occurrence of cancer. Certain times, if it is a localized cancer, we remove that cancer, and what we try to do is there should not be any relapse. So for that also there will be a continuation of treatment will be there to that particular patient. Okay, so how should the impurities in the drug be managed? So in such cases, so initially the drug was meant for small duration treatment and advanced cancer. Now the drug is for long-term survival of the patient is surviving around 2 to 3 years, and for different type of cancer. And how to because here in the first case, no, even there is a small percentage of impurity, still will allow the agency, either ICH, FD or WHO, whatever the EMA agency will be there, no, they will allow, we'll go for risk benefit because we want to keep the patient survive. Small amount of impurity is not going to cause immediate threat. The immediate threat is here cancer, but here continuous exposure to the small impurity, and that impurity itself is genotoxic and impurity itself is causing cancer, then that will be a cause for a potential concern. Then you have to do risk assessment of that particular impurity. So what is the answer which is given by the ICH agency? Will see. So when an anti-cancer pharmaceutical is further investigated in cancer patients population with long expected survival, for example 2 years or 3 years, ICH Q3A, Q3B, ICH M7 should be considered for the control of impurities. So if you go back to the first page of ICH S9 guideline, very clearly it states this is only applicable to those cancer treatments which are in the severe stage. Okay, so short term, immediate life threatening, so other type of cancers, long term survival cancer patients, you have to qualify the impurity scenes for such anti-cancer drugs. You have to follow Q3A, you have to follow Q3B, and you have to follow ISHM7 guideline. So I would request you to please go and read these ISHQ3A guideline R2, ISHQ3B R2, which is for this is for new drug substance and this is for new drug products, and ISHM7, which is coming in 2023, and this is the main thing for mutagenic impurities. Okay, assessment and control of DNA reactive mutagenic impurities pharmaceuticals to limit potential cancer risk, ISHM7 R2 guideline. Okay, so in this particular video we have discussed other considerations under ISH S9 question and answer document. Try to understand whatever the new drugs which are coming into the market, a larger section are on the anti-cancer drugs. So ISHS9 guideline is a dynamic document. It keeps changing because new information comes, new type of targeted therapies are coming, so which are not addressed under the current guidelines. So a lot of revisions are taking place, a lot of new queries are coming from the pharmaceutical industry. So some are other questions they will ask with respect to ISHS9 guideline. So that is the reason I have discussed in detail all the question Q&A document before guideline coming into as a publication, no, it will put it for the industry, so that questions will be asked by the different pharmaceutical companies, and that will be addressed by the ICH agency. Then after series of discussion, then finally a guideline will be done. So this is a pre step before the guideline. So some of the questions which are not addressed in ICH-S9 guideline, main guideline, can be addressed under Q&A document. Okay, so we have discussed in detail, whether we have discussed about ADC, linker, pay linker, what type of tissue distribution studies we have discussed, half-life, plasma stability we have discussed in this section. We have discussed about impurities, genotoxic impurities, ISHM7 guideline, how to address these impurities. Okay, so and we have discussed about this one also, FIH related questions, STD as well as HNSTD. Okay, so thank you so much. Be curious. For any questions, you can always write a mail to ra.priyanka18@gmail.com. All the best for your exam.